All right, everyone, let's get started. Can someone confirm you can hear my audio? Thank you, Mariana. So, hello, everyone. My name is David Ruck. I'm here at the University of Maryland in Baltimore, Maryland, and welcome to Cytometry in R. So, this is the second week of the course, and we'll be talking about file paths, which is basically how do we communicate to our computer where to find our particular set of FCS files so that we're then able to use them. So a little bit of bookkeeping. If you are just starting the course, don't worry, you're not too late. There's still plenty of time to catch up with where we're at currently. All you really need for this particular lesson today is having set up a GitHub account, as well as having installed required software, which is covered through the workstation setup walkthroughs. If you have been able to do the pre-course walkthrough for how to use version control with Git, that would additionally be helpful, but that's not required. We'll cover the pieces of that we need today throughout the lesson. So when we're first starting off trying to learn how to code as flow cytometrists, we have big dreams and we try to think in very big picture. We want to take our files, we want to normalize them if we're going to do some unsupervised analysis, we then want to cluster them, we then want to analyze them, and finally we want to plot them into pretty pictures we can use for publication. So when we first try to write code, we tend to plan and write our code in very similar broad strokes. By contrast, our computers don't really think in big picture, but they think more in small incremental steps. So while you're thinking, let's normalize this, let's cluster this, your computer's very much still on, let's first locate this folder, let's list the files that are present, let's select specific ones, let's import them into R, let's then load in the normalization package, let's pass the first file, etc., etc. So all these little steps combined algorithmically add up to creating the bigger picture. But when it comes to trying to problem solve and troubleshoot something that's not quite going as expected, we have to really think how our computer is thinking in order to be able to solve these issues. And it's not like as immunologists, as flow cytometrists, we're not immune to being able to think in small pictures. So for example, let's think of we are doing an experiment. We don't just think of it in terms of we thaw some cells, we stain some cells, we go upstairs and acquire them on the flow cytometer. If we just break down the individual steps after we thaw the cells, we need to spin them down in a centrifuge that has a certain speed and a certain amount of time. Afterwards, we dump blot and vortex the cells. Then if we go do a live stain, we add a live dead, etc., etc. So we can also think of, just like we think of our own experiments in like small incremental steps, we just need to learn how to apply this small incremental step thinking when it comes to working with our computers and coding in R for the very first time. So when we are first starting off, file paths tend to be the first place we end up clashing between our big broad goals and the actual coding mindset that's needed to communicate to your computer. And this is mostly because we're often working with experimental FCS files. They're stored on our hard drive, they're stored on an external USB that we have plugged in, and we can clearly see that the files are there within a folder. We are looking at it generally within our file explorer, depending on if it's a Windows, a Mac, or a Linux computer. And then you're just like trying to communicate to R how to find this file so that you're able to do your analysis, and it's just not cooperating. So for today, the main goal is to kind of clarify what's happening with working directories and file paths, but then also we need to introduce some basic R programming knowledge in terms of how to create objects, also known as variables, how we can name these, how we can assign particular types of values to them, as well as also be able to like look at a list, get things back that are in a certain position, etc. So this would be stuff that if you're taking a more traditional basic R course, it would go on and on about types of objects for like a couple of weeks in a row. I'm going to try to make this a little bit more interactive, but you still need some of this basic knowledge if you're going to be able to do. And it will be helpful later on today when we're actually going and trying to do work with file paths as far as being able to assign it to a particular object and then bring back in those particular file names later on. So Before we go and start talking about file paths, I want to kind of like walk you through what I normally would do. Like each week of the course, we'll be giving you new data sets, there'll be new code to bring in. And I want to make sure more than anything that you're able to quickly and efficiently bring in those new data sets to your own computer. But then I also want to make sure you are storing those data sets in a different location that you can work with. So no matter what you're doing, if you're renaming a file, if you're changing its location, it doesn't cause any merge conflicts next week when you try to bring in the next data set from your Cytometry and R folder. So this is also kind of my workflow I will do for just random projects I'm working on. So if I have a coding project, I will do kind of these similar steps. So this is just a good walkthrough of like, what do you do to try to keep your code organized, your projects tidy, and be able to like make sure like if I need to transfer this to my supervisor later today, I'm able to transfer that file, the project folder, and everything within the code should still be able to run. So the very first step, if you're doing this like every week for the course, is I want you to create a new repository. And we've done that already in the workstation setup as far as going to GitHub. And when you go to GitHub, there's that little plus sign that's on the upper right side of your screen. And if you click it, you can see the drop-down menu, and there's the option to create a new repository. So you can go ahead and create a new repository for each week. Basically, what you need to do is just make sure it's a project folder that you're able to work with and you're able to customize. So if you want to call it week 2, week 3, week 4, whatever you want to do, that works. If you just want to call it my testing folder, that's also fine. The name of a repository is very much up to you. And because this is more you're working with stuff you still are not quite ready to share this code with the rest of the world, we're going to go ahead and set this repository to private for the time being. So it's like if you look at my own GitHub account right now, there's like 20 or so projects I share with the rest of the world. Then if you like look at my particular view of how I'm able to see all my code, there's like 250 or so private repositories where I have like particular projects worth of code. Not some of them are not ever going to see the light of day, but I have them organized so if I ever am blanking of like what did I do for this particular project, I can always go to GitHub and see what the code is there, and if I need to, I can search for a particular line of code that I need. And then in this case, because this is going to be one of your own repositories, it doesn't matter whether you want to add a README file, if you want a git ignore file, or you want to do a license. We'll talk about those more in context of when we do a public repository. So once you have created your new repository, what you first need to do is copy the URL. So if you're in GitHub, it'll pop up that new repository window. You just come in here and copy the URL, at which point you need to navigate back to Positron, and kind of similar to what we did for one of the pre-course walkthroughs, right next to your interpreter, which is where you select to use R as the programming language for your particular session, you need to go to this little folder icon and select the drop-down, and then you're going to select the option to bring in a new folder from Git. And when you do so, basically what you're going to do is copy and paste in that URL for your new project folder. And then wherever you've determined you're going to be saving your R project folders, go ahead and save that project folder to that location. So in my case, for across all my computers, it's always in Documents. It doesn't really matter where you have it. I just emphasize try to keep it on your local computer. Don't try to keep your coding projects on a remote location like Dropbox or OneDrive, just because they can have permission issues, which can cause some weird bugs that you won't be able to troubleshoot. So just as long as you store it local, at that point you should be good, and go ahead and hit open. And at that point, what I do for every single project, regardless of what it is, I try to keep some basic infrastructure organized. So if you come into Positron and you look at your left primary sidebar, you see you have this option to create additional folders within that window. So I'll just go ahead and create a couple additional folders. I normally will create a data folder where I'll stick any FCS files that I need for that particular project. I'll create an image folder just in case I'm taking screenshots and I want to document what I'm doing. And I'll just create an initial Corel Markdown file. In this case, I named it file path using this new file option just so I'm able to start writing code and documenting what I'm doing. And then at that point, we are pretty much ready to go as far as getting the initial parts set up. Now, if you are trying to bring in the new data sets that I upload either on Sunday or Monday each week, and bringing in all those code changes that have happened since, as far as me updating the slides, updating things when I catch some typos, what you need to do is you need to go back to GitHub, where you have your own forked version of the Cytometry in our folder. So if you're up to date with all the changes I have made on the actual course repository, you'll get a sign that kind of looks like this, telling you your branch is up to date with the parent repository. But if I have made changes, uploaded additional data sets, etc., you'll see this branch is behind by several commits. Do you want to update? In which case you'd go and say, yes, I want to sync to fork. Once you have synced to fork, all the data is now up to date on your GitHub account, which is remote. but you still need to bring those changes down to your Cytometry and R folder on your local computer. So at that point, what you need to do is you need to switch project folders within Positron. And the way you do this is basically if you go back up here and click on that folder on the upper right side of the screen, you can see we're currently within week 2, which was the most recent thing open. But basically, you need to go back across the different projects that you have been working on this week, and then basically select the Cytometry and R forked version that you have already on the local one. And at that point, Positron will reopen, and you'll be in the Cytometry and R local version of the folder. And at that point, to bring in all that new data that's coming in for a particular week, you just need to go to the Actions bar on the far left side of your screen, select that Source Control little tab that's there, and then you'll go to Changes, where you see the ... It'll give you the drop-down menu of options for version control, and you'll just select this Pull option, at which point all the data sets that are coming in for this new particular week will be brought into your actual computer, and you'll be able to use them. So in this case, for this particular week, we're working with a data set of 8 FCS files. They were used to estimate what the cell concentration was for some cryopreserved specimens that were both adult donors as well as cord blood mononuclear cells. And I have significantly downsampled these. They're only like 85 kilobytes each, so very, very small. They're only like 100 cells in each of these files. Within the organization of the Cytometry and R folder, everything will always be under course, and then for the particular week. And then if you're trying to download the data set for that particular week, it'll be within that data folder. So all you'll need to do for today, if you're trying to follow along, is basically you'll need to copy these 8 files from their current place in the Cytometry and R folder. And I want you to go and paste them into the week 2 folder within that data folder. So that way you can do any changes that you want to those particular files. And no matter if they get completely corrupted, they're not going to affect anything that happens within the Cytometry and R folder. So you'll still be able to bring in those files throughout the next couple weeks. At which point, if you go back and reopen the week 2 project folder by going to the upper right and selecting the respective project folder, you'll get a view that looks kind of like this. Once you pasted in the 8 FCS files to the data folder, you'll see they're being tracked now by version control. Go ahead and actually stage all those files of interest and go ahead and write a short commit message. This way, you're starting off fresh. You have all the data that you have now set up, all those project folders. You have now all those little folders within your project folder to keep things organized now set up. So at this point, this is a good stopping point as far as setting up a waypoint. In case you do need to revert back changes later on, you won't be losing too much effort as far as getting your initial project set up. So this is kind of what I will do for every single week as far as like keeping my projects organized, but then also like following along on the course. Just keep everything that you're doing as far as active code editing to one project repository of your own making. And then at that point, once you have committed everything and have it as a commit, just go ahead and send all those changes you made to your own project repository back to Positron, back to GitHub, so that worst case scenario, something happens to your computer between now and lunch, you would still be able to have those changes, that code data sets that you have made available on the remote GitHub. All right. And with that, let's start talking about how do we communicate to our computer where to find things and how to locate where you're at currently. So within R, we have what's known as a working directory. So the way you get at this location, and you're kind of like how your computer perceives where you're at in the active moment. So we're going to be showing quite a few functions today. I will be introducing them, and just to remind you, functions are particular tools that do a particular task. And their big giveaway in R about what a function is you'll see these little parentheses with the function name to the left of it. So in this case, we'll be starting off with the getWD function, which basically stands for get your working directory, which is going to return the location of the folder that you're currently within as far as how R and how your computer view where you're at in the current moment. So if you're within your week 2 project folder or the equivalent that you have named, if you run the getWD command, you're going to get back a return that looks something like this. So the first part of this, the actual address to get to your folder, will vary depending on if you're on a Mac or Linux or a Windows operating system. So in this case, you can see this is within my home folder, within David, it's in my documents, and we're currently within the week 2 folder. If I was on a Windows computer, it would be something like C, users, my username, documents, and that's how the upper portion here would be showing up in the file path itself. But as far as your computer is concerned, it's not aware of what's happening up here. It's only focused on that particular folder, week 2. So what your computer sees is basically anything that's right adjacent to the week 2 folder. And this is what we'll know as your active working directory. So when we're working within your working directory, if we're looking within the positron window, we know we just created this project. We know we just created 2 folders, data and images. But we can also get these things returned individually by true_r by using the list.dirs function, which will basically give you back a list of the directories that are present within a particular location. So if we were going to write out this actual function name, it would look something like this. If you are doing this in positron, if you're writing the function within a chunk of code, if you hover over it, it's going to open up the help documentation. And then you would see like the actual arguments as far as what these particular arguments do be available there. So in this case, we'll go into what each of these arguments does in just a little bit. But basically for now, we're setting the path is equal to quotation marks period, which is going to stand for our current location. Full.names, which is a particular argument, we're going to set it to false. We'll talk about what it does in just a bit. And then the recursive argument, we're going to also set that to false. So when we set a particular argument to be true or a particular argument to be false, what's basically happening is we're telling the particular function we're interested in this case, list.dirs, either don't do this when it's set to false, or do whatever this argument is specifying when we have set it to true. So if we go ahead and run this particular line of code within our working directory for our week 2 project folder, you'll see we'll get back 3 folders being listed. There's a .git folder, which we'll talk about in just a minute, which we can see is not showing up in positron. There's also the data folder, which we have just created, as well as the images folder. So as we are looking at this, our first argument we were working with was that path argument, which we had set to quotation marks period, which is basically just a stand-in argument for our present directory. Now, if we had not included this argument, R will still kind of infer, based on its particular arguments, where it should be grabbing the location from. So it's kind of defaulting back to your working directory. So if I didn't include the path argument within the parentheses, you're still going to get back a very similar result, showing you what's present within your working directory, meaning mostly the data and the images folder. So within positron, in addition to those visible folders, we also have what's known as hidden folders. And these hidden folders are denoted by having a period in front of the name. So in the case of the website for the course, if you look at it a little closer, you'll see that we have like a .cordal hidden folder. And this is basically, if you're looking at it within positron, shown in a lighter type of gray. These typically are files that are more involved in like how you're seeing things displayed on your screen within the software. They're not stuff that's actively involved in your code. So these are going to be hidden as far as like you're not going to be actually seeing them unless you want to see them. The .git folder is by positron hidden by default just because it's one of those things that you will not be doing individually most of the time as far as adjustments goes. But just to let you know, it's there, it's just hidden in the background. So in the case of week 2, we had those 2 not hidden folders that we had just created, the data folders and the images folder. So we'll be navigating to these and trying to figure out how to tell R we want to copy things to those folders or we want to check and see what's within these folders. We'll circle through that one in just a minute. However, before we do so, we should talk a little bit about some basic R objects. Basically, how do we create a particular object or a particular variable? So when we're doing so, in R, we have the ability to assign a particular value, whether it's a character string, like a name of a floor, a particular number, like the number of floors in our panel, or a logical, like is it true or false, to a particular object, which is also known as a variable. These can then be used when we need them later on, whether it's several minutes from now, a couple hours from now, to provide that variable back to our lines of code. So a simplest example of this is if we want to provide something, we could say, I want to provide a variable, a particular value, which will return as Saturday. So since this is characters, I'll have to include the quotation marks on each side of the word. But then you'll see this particular lesser than and hyphen right next to each other. This kind of works like an assignment arrow in R. So basically what I'm saying is, for whatever my variable or my object name, which is basically whatever name I decide to give it, that's located on the left side of the assignment arrow. In this case, my object or variable name is going to be what day did I write this. I'm basically saying For this object variable, what day did I write this? Assign it a value of Saturday. At which point, once we have run this line of code, we'll see if we look at the right sidebar in Positron, within this session tab, you'll see the variables. This has now created an object. It has created that variable, what day did I write this, which now has a value of Saturday. And then at that point, you can use it later by just calling the name of the particular object that you have created. So if I then go back into my console window or within a code chunk and just write out what day did I write this, and I hit enter, R is going to search within its environment. It's going to see that that object, that variable is present, and it's going to return you the value for that particular variable. So you can create and name a variable however you want to name it. There's no particular hard definition as far as how you should be naming a variable. The main goal of when you name something is you want it to be memorable enough that you know what it does, but then that other people can infer if they're reading your code what it's particularly doing. So in this case, let's say I didn't want to do what day did I write this, but I want to create a variable called top secret meeting day. I can also create a variable called top secret meeting day and give it the same value for Saturday. And at that point, it would appear within the variables drop down once I have created it. There's a couple exceptions to this rule, though. R does not play well at all when it comes to spaces. So if you can avoid using a space in R, I would recommend doing so. So in this case, you can see I tried doing the exact same sequence of words, top secret meeting day, but I kept the spaces in between. At which point, when I tried to assign the value of Saturday to it, it did not work because at this point, R got confused. It tried to assign it at top, but then it didn't know what to do with the next couple words in the sequence, so you got an error at this point. However, if I was trying to name a variable, but top secret meeting day, and I still wanted it to be able to be a little bit more readable, I could have added underscores, top underscore secret underscore meeting underscore day, and then assign Saturday to that, and that would have worked perfectly fine as far as an object name. So when we're putting those underscores in between each word, we're basically using what's known as snake case. I will tend to use what's known as camel case, and the best illustrative of what I mean by this is kind of this illustration I found online. So different bioinformaticians, different software developers will have preferences as far as how do they go about naming the particular objects or the particular variable names. Some, if they're using like multiple words, will separate them by that underscore, which is what we kind of know as snake case. Some of them will use capitals for every start of every other word. So in this case, you'll see it kind of looks like a hump of a camel as far as when you're looking at an appearance. And then there's others less used, like screaming snake case, kebab case. There's a lot of quite ingenious names when it comes to figuring out how to describe what you're doing. The main goal ultimately when you're creating or naming a variable is you need to remember what that particular variable does, what particular kind of value it will have, which means if you make it more descriptive, the better off you're going to be. But then it also still needs to be interpretable by somebody else who's trying to read your code. So in this case, if we go back and look and see what we had top secret meeting day giving us back a value of Saturday. But in R, you can also overwrite a particular value that's assigned to a particular variable. So let's say we don't want our top secret meeting day to be Saturday. Let's say we're trying to switch this to Monday. Basically, we're just providing the new value and reassigning it back to that particular object, at which point, if we then are able to just print the object name, we see that the variable has been changed. It has been updated to Monday at this point in time. We can also remove variables that we have already created. The way we do this is with the remove function, which is the rm parentheses. So if you had a particular variable, or if you created an object that's very, very large, like it's gigabytes in size, and you're trying to get rid of it from your local environment, you could use rm parentheses that variable object's name and get rid of it from that particular session. However, if you're also working within Positron and you're looking at the session of variables, you can get rid of individual ones by deleting them here, or if you're just trying to delete all the variables from the session, you could go up here and just delete all the actual created variables. So in that last case we've been working with, we were working with what was a string of character values, basically Monday, Saturday. Everything there is a letter that's present and surrounded by the quotation marks. So if we are thinking about it, there's going to be different types of variables or different types of values that we assign to our variables. There can be numerics, there can be logicals, which are either true or false, and we can also have characters. How do we go about seeing what kind of type is assigned to a particular object? And this is where the str function, which kind of stands for structure, comes in. So in this case, if we look at this one here, we can see I'm creating a new object, a new variable. I'm calling it floors, and I'm assigning Fitzy to that object. So if we rerun str for floors, it'll come back and then gives us back the value that's present within floors. But we'll also see it's giving back the type, telling us that it is a character object at this point. We can also do this in a different manner because like in R, everything that you can do one way, there's at least 2 or 3 other ways that you can get back those exact same values. So in this case, we can also do it with the class function. So if we run class on floors, it's going to give us that this particular value assigned to this object is a character value as far as what we're seeing. And then we can also see how this differs is, for example, if we had to assign a numeric value to a particular object. So let's say we have been talking about how many markers are in our panel, and we had to assign 29 to floors. If we call str function the floors object, we'll see that it comes back at this point as numeric, and it gives you the value. And similarly, if we had assigned a value that was true or false, we can then check with str and see is it a logical type object. So in this case, I'm making a new object or variable, which I'm calling is Percy Sci 5.5 a good floor, and I'm just going to assign a value of false to this object. If I then call str and then go ahead and double check, you can see it's coming back as a logical as far as the value within this particular object. So, and thinking back to like what we did last week, we saw that error when we tried to install the plier, but we forgot to include those quotation marks. So what we just covered kind of explains what we were seeing. So basically, when you don't have those quotation marks, which kind of make it a character value, R is thinking it's an actual object or an actual variable that's already present in your environment. So that's the reason why when we use install.packages, which was the CRAN-specific install command, but the plier without any quotation marks around it, it was looking for an object or a variable called the plier. It wasn't finding it within your local environment, which is why it was erroring out. However, we could have done a slightly different approach that would have also worked. So for example, if we had specified the packages that we want to install is quotation marks the plier and assigned that to a particular object package to install, and then just use that object, that variable within the install.packages function, it would have checked for that object, it would have seen it in the environment, it would have provided the dplier value, and then it would have installed without any issue. All right, so we talked a little bit about how to find our working directory, and we've talked about how R goes about naming particular objects or variables. We don't always work with just a particular object or variable that's just a single item present within. So, for example, let's go ahead and modify our floors object. So instead of just having it as being Fitzy, let's add a couple additional floors. So let's add BB421, let's add Fitzy, let's add PE, and let's add APC. So if you look at this particular line of code, you'll see this little lowercase c next to parentheses. This basically stands for concatenate. So instead of just being able to provide a single value to floors, if you put C parentheses and then each of those character values we're trying to add into the floors object with quotation marks and commas in between them, it's going to basically create what looks like a list, which is known as a vector in R. And then at that point, if we come back and check what's within floors, you'll see it actually has positions. It shows you that there's 4 objects present within floors. 1, 2, 3, and 4, and then it lists what's actually present as far as BB421, Fitzy, PE, and APC. So this actual like listing of position is what we know as indexing in the context of R. So we can then check to see like is a particular object a single value object or does it have multiple values present within? The way we do this is with that length function. So if we went back to floors and placed it within our length function and checked, we'd see that there's actually 4 values present within floors. And now that we know that there's multiple values present within a particular object, we can index them by using square brackets. So, for example, if we provide the name of the particular object that we're interested in, and then within square brackets provide the index, basically the position of the value that we want to see, we can retrieve those values individually. So in this case, floors1, based on how we had created it, is going to return BB421. If I wanted to check what was the third object in the floors vector or list, we could see that that value was the PE value. If we wanted to return multiple of these, we'd basically use a colon. So this is what it would look like. So basically what we're telling R is, for this floors object, grab everything from the third all the way to the 4th object and return those values to me, in which case it's returning PE and ABC. And those values don't necessarily need to be right adjacent to each other. So if we want to go back and reuse what we learned about the concatenate function, you can kind of see, we can basically say, hey, I want to return the first value and the fourth value of this fluorophorus object. So this kind of works within those brackets to index it, which returns basically the first and the last value that we had assigned. So it returns to us BB421 and ABC. So this is kind of how indexing works in the context of R. We'll go through this a lot more throughout today when we're talking about returning individual particular files that we may be interested in. All right, now that we have some of that basic R knowledge as far as how to create objects or variables, how to name them, and then a little bit about how indexing works in terms of a vector or a list, let's go ahead and go back to our Week 2 project folder and try to retackle figuring out what's present within our data or images folder. So the first function I want to introduce is basically the one we've covered already. We use the list.dirs function to basically return all the directories or folders that are present within our working directory. So in this case, we saw we had data and we had images. Now, there's another function that's very similar, the list.files function, that will return to us not just the folders, but all the files that may be present within a particular folder. So in this case, if we run list.files, you'll see that within our working directory, we have both the data and the images folders. We also have our file paths.qmd folder that I had just created. And then we also have the license and the README files that are created when we set up our GitHub repository. So this is just listing the files that are present within our active working directory. By contrast, if we were interested in our data folder, which at the moment should only have the SCS files we copied in manually, we can basically adjust where we're having our computer look. So basically, instead of not providing a path argument, we're now providing a path argument and giving it the name of a particular folder that we want to go and look into. So in this case, basically, we're telling the list.files function, hey, look within path, this particular location designated by the path argument. So in this case, we're telling it, look within data, and then give us the list of files that are present, don't include the full names, and don't follow this recursive argument. And at that point, it's going to return those 8 files that we have already copied over into that data folder. And we're going to be able to see the file names of these particular files. So at this point, the only thing within our data folder within the Week 2 project folder we had just set up, there's no additional smaller folders underneath. So in this case, I'm just going to go ahead and create a new folder and place it under data. So if you're working within Positron, you go up here to that little data folder, create it, name it as target, and then you should be good to go. One thing I'll mention is if you accidentally create a folder and it's in the wrong location, let's say it's down here, you can just click and drag it up to place it in the correct location, which is very helpful. If you decide you don't want a particular folder within Positron, you can just click on it and then hit delete, or right click and select the delete option, and that will also get rid of a particular folder. It works much more similar to what you're used to on a computer than what we had previously for RStudio. So at this point, we have created a new directory via Positron, but we can also do so via R. And the way we do this is basically we need to provide a location to the function that's known as dir.create, which will basically create the directory for you in that particular location. So in this case, we need to specify a location, so we'll use a function that's known as file.path, which we'll revisit quite extensively later today. So we're basically saying, hey, we want to create a file path, basically an address that includes the data folder and the target2, which is going to be that new folder we're going to be trying to create. So at this point, if we run this together, it's going to create a new folder location variable, which will just basically say this particular address, data/target2, is what you are looking for. So when we hand that to that dir.create function, it's going to see if there's already a directory present. If there's not, it's going to then create the directory at that particular address, which will result in us creating a target2 folder. So in this case, if you run that command, we'll see now we have a target2 folder that's present within our data folder. So Before we continue, it's not very useful if we have two folders that don't actually have objects within them when we're trying to list the objects that are present in those folders. So I will go ahead and copy over two of the FCS files from the data folder to these subfolders target and target2. So you can see I copied and dragged them. Version control has now shown that these are new files that are appearing within these folders, so it's tracking our changes over time at this point. So at this point, we have looked at our working directory, and it's been entirely focused on what's happening within that week 2 project folder. But R does not actually know what's happening within those individual target folders by themselves. So for example, if we try to rerun the list.files command, but we just provided the name of the folder, target, it's going to come back with a character equals 0, which is basically a it did not run successfully output. And the reason for this is if we want to see what is R actually seeing, is it know that the target folder actually exists, we can run a function called file exists. And if we provide that value for the target folder name, you're going to see this comes back to us right now as being false. By contrast, if we wanted to check to see if the data folder exists as far as our computer and R is concerned, you'll see that the data folder is returning a value of true. Now, the reason for this is our computers only know what's happening within the working directory and maybe one level down from that working directory. If you're working with something that's a nested folder, that's within a couple folders before your working directory, you're going to need to provide a full file path to that folder so that R knows, hey, I don't need to just search within my working directory. I need to go search in this particular location so I can then index the files and return a list of the files that are present. So in this case, there's a couple ways that we can do this. When I'm specifying a file path, we showed that example a little bit earlier where we did data slash a target. That's basically specifying what we already see from our file explorer as far as each folder being a particular argument that's then separated by a slash. Now, whether using a forward slash or a backward slash depends on which operating system your computer is running. So macOS and Linux will do forward slashes. Windows will typically do backward slashes. This is something to keep an eye on if you're planning to share your code with others. So what this will look like if we just wanted to fix list.files for this particular application, just so we can see which FCS files have we copied over to the target folder. If you're working on macOS or Linux, you just add a slash between the data folder name and your target folder, and then at that point it would run successfully because your computer now knows from your working directory, check this location, this particular file path, and then return the list of files that are present at this particular location. However, in this case, what we're doing is we're providing kind of a hard-coded slash, and this can cause us some issues, especially if we're working on multiple computers that have different operating systems. Because at that point, we're basically assuming the next person who's going to try to run this line of code is using that same kind of operating system as us. So it's going to be a hard-coded slash, but then let's say if I'm suddenly using Windows and I try to use a code that was set up for macOS, whether a folder is using the backslash or the forward slash to separate out individual folders is going to be different, and we're going to come back with an error. Fortunately, we have a workaround that is quite useful and kind of takes out this thinking part from our actual day-to-day responsibilities, and that is the file path function that we've been working with a little bit already today. So basically what file path function will do is at the very start of it's evaluating a line of code, it's going to figure out, are you on macOS, are you on Linux, are you on Windows, and then it's just going to insert the correct forward slash or backward slash for you without you actually having to worry about it. So if you're using file path to designate a particular location, what you'd basically be doing is for each of the folders from your working directory down that you're trying to specify, you just include the folders within quotation marks because they are character values. And then at that point, it's going to create an object, which is a file path. So if we call folder location in this case, we can see it returns a file path of data that target. If we were on a different operating system, it would switch the forward slash for a backward slash just to keep it compatible. So then that way, you can transfer your code to anybody across any computer system, and it's just going to automatically update the file paths to represent what's actually happening on their computer, which is quite good when it comes to reproducibility and replicability. And like we see in this case, using this particular setup, we can then provide this object or variable name to the path argument, it then goes and checks on our local system for the object or the variable called folder location, and then list files does its thing on that particular path location to return the particular files that are present. All right. So we don't always have to completely write out all our file paths. If we already have an existing file path, we can just append that object inside a new file path argument to kind of extend it out further. So, for example, let's say we had set folder location just to a single path, location being data. We can use that object or that variable within the file.path function, and then just append on the new location we're trying to search for, and then assign this to a new target, a new object, in this case the screenshot folder. And then at that point, it's going to just append on as we go. Additionally, list files has a very useful feature. It has an additional argument we have not been using already, and this is the ability to filter files that contain a particular character string. So let's say we're only interested in CSV files that were in a particular folder, or we only wanted to see our FCS files listed for a particular folder. Or let's say we're working in a folder that has all our single color reference controls, and we just want to get the APC size 7 out of all those different FCS files that are present. We can basically use what's known as the pattern argument, which if you provide it a character value between those quotation marks, it will search all the things that are present within that particular folder and only return objects that have that particular character string somewhere within the file name. So if I ran it, for example, on the screenshot folder, which I had just designated for the object, it will only return the adult specimen because it's matching the ND050 character values that are located right here. Now, this pattern can be whatever we want it to. We could have filtered based on cell counts. We could have filtered on .fcs, as long as we included it between the quotation marks. The files for today were acquired on both a 4 and a tree laser Aurora. If we wanted just the tree laser, we could have used the tree laser's code designation as treeL, and it would have only filtered for those particular files that we were interested in. So you'll also notice in this case it only returned a value of 1, even though that original vector that we had back was it was a second location for the ND50 specimen. Listing of files is kind of dynamic. It only will index based on what is actively returned. So in this case, it's returning as the first object with the first value within the actual object we're creating. All right. Next up, we're going to be talking about how to select for patterns. We're going to go a little bit more as far as looking at a couple extra examples. So far we've only been looking at files that are listed within data, and we get returns back that look something like this. So if we ran it again on data without any pattern argument, we can see within our data folder inside our working directory, we have all those FCS files, but then we also now have 2 additional folders. We have the target folder and the target2 folder that we have just created. So let's say we were trying to pass these objects to the Flowcore package, which we'll be using starting next week, and it's going to only be handling FCS files. If we hand it a list of objects that also includes folders, it's going to run successfully for these first 8 FCS files, but then when it gets to that 9th object, it's going to error out because it doesn't know what to do with a directory folder. So we kind of want to be able to subset down this object of list, this list.files list, just to include what we actually want to be using for a particular function. So we already saw when we looked at it in context of list.files, we can specify a particular pattern. So if in this case we're only interested in the .fcs files, we can provide that matching argument for pattern is equal to, between quotation marks, .fcs. In this case, we can assign it to a new object which we're calling files. And sure enough, at that point, we have removed out those 2 folders that we don't actually need, just keeping the actual FCS files that we are actually interested in. Now, the pattern argument is working within the list.files function, but as we go throughout the course, we're going to need a little bit more complicated character strings as far as being able to match objects. So what handles this in R is the stringer package, which is from Tidyverse, which I had you install starting last week. And within this package, there's a function that's called structure detect, which has a lot of the same functionalities you get from this pattern option, but it also has a few additional things that would make it quite useful. So in this case, let's say, example, instead of the adult PBMC specimens, we wanted to look at the infant CBMC specimens. We could basically have structure detect look within those files that we have listed and see if it can find the code INF, which is standing for infant, and then basically tell us, is this particular file on the particular index, i.e. the particular location, a infant file or is it an adult file, and give us back true or false values. So if we wanted to actually run this in code, first we need to load stringer by the library call, because we need to attach it to our local environment so that we're able to access the structure detect function that's within. We don't want to repeat my mistakes from 2020 in this case. And then at that point, what we can do is, after we look up the help documentation for structure underscore detect, we see that basically we're providing that list of files, in the case of files, as the first argument. And then our second argument is basically going to be the character value that we're trying to match it to. So in this case, you can see it returns back true, false, etc., etc. What it's basically doing is, if we go back and look at files, you can see that very first index does have INF present, so it's coming back as true. By contrast, that second index is a PBMC specimen. There's no INF pattern present, so it returns as false. On the third index value, it is INF present, so it comes back as true, etc., etc. So at that point, that command that we just ran is basically telling you, is this pattern present for that particular index in a particular object? So while this may not be particularly useful individually, we can kind of combine that with the indexing we did already previously with the floor from 1 to 2, because basically if we include those true-false values that are equal in the number that we see within that actual object, it's only going to keep values that are true present, and it's going to get rid of all those values that are false. So if we try this right now in an example, we know that structure detect all the way through the end of the parentheses is going to give us back true, false, true, false, true, false, as was determined by the character matching. But now that it's placed within those square brackets, it's only going to keep the values that are true, and it's going to keep those particular index values. So it's going to basically say keep 1, keep 3, keep 5, etc., based on where the true or the location was located. And at that point, we can see we have subsetted, we have filtered at that point for just the infant files that we are particularly interested in. So in this case, we have just gone ahead and indexed all those files as being just the infant CBMC specimens. Let's go ahead and save those to a new object, a new variable name, which we'll call infants. All right, and before I start on conditionals, let me take a sip of coffee. All right, so at this point, we've talked working directories, we've talked variables and objects names, we've talked about indexing, and we've looked a little bit at the file paths arguments as well. So one very useful thing about R is we can set conditions on whether a particular line of code gets carried out. So the most typical conditional that you'll encounter in your daily practice is going to be if statements. And if statements look something like this. So if statement starts off as if, and then there's a parentheses, and then there's some line of code that's present within that parentheses. If whatever line of code within the parentheses comes back as true, the if statement is fulfilled, and then at that point, any lines of code that are between these two curly brackets will actually be run. So in this case, let's say within the parentheses, we have an object or a variable which we called as need coffee. And let's say we have set the value of need coffee equals to true. In this case, need coffee comes back as true, at which point the if conditional is satisfied and is allowed to run. So all this code that's now present between the print and between the curly brackets is now actually run, in which case it runs the print function, and you'll see it actually displays the text that's present within your console window. By contrast, if we had set that need coffee value to false, within that if statement, this value returns as false. That condition for the if statement to run is not met, and therefore we don't see the line of code between the curly brackets get run. And this can be quite useful, especially like if you want to work on something that is a raw file, you could set up a conditional statement basically saying, if you identify this as being a raw spectral flow cytometry file, first go ahead and unmix. Versus if you're running a list of files that have already been unmixed, it would just ignore all the steps that happen with the unmixing and would just proceed to the very next one. And you can think of a lot of different examples as far as like when you may want a particular set of line of code to run, but not happen in a different context. So this is kind of the options you'd be able to do if you're using a conditional. So the if statement will trigger as long as whatever you have within those parentheses right after the if, that condition is met. So for example, let's do a numeric example for this one here. So let's say we assign the value 299 to the object we're calling as row number. So within row number, what we have is 2 plus row number is a value greater than, it's 301. And then 301 is going to be greater than 300. So in this case, if we evaluated this line of code, it would be coming back as true. So since this value is true, if we place that line of code present within the parentheses, it will then actually evaluate the statement and would see if this value is true, which it is, it's then going to be able to print out that particular line of code that's within your curly brackets. One thing I want to introduce, just because you will see this at some points throughout the course or just reading through general lines of code, if you add an exclamation point in front of a conditional, either meaning something that's true or false, a logical value, it will basically flip what your expected outcome is. So for example, if we're working with an object or a variable name we're calling it's raining, and we assign it a value of true. In this case, if we looked at this conditional statement, if we looked at it, it's raining is going to come back as true. So this conditional statement has now been fulfilled. So it's going to actually run the line of code that's within these curly brackets, and we'll see bring an umbrella gets printed to our console window. By contrast, if we just look at our actual object or a variable we're calling it's raining, and we add a little exclamation point in front of it, we can see that this value is now flipped, and instead of being true, which is what we assigned it, it's going to be read as being a false value. So if we had instead placed the little exclamation mark in front of the it's raining value, even when we have this set to true, it's raining is true, it's going to be read by that conditional statement as being false. So in this case, it won't actually trigger the line of code between the curly brackets, and you'll never see that output being displayed. And we'll show you where this may be useful later on. It's particularly useful if you're trying to create a folder that doesn't already exist. All right, so conditionals can get more complicated, and we'll see these throughout the course. So for now, let's just focus on a couple simple ones in the context of trying to copy over our FCS files to a new target folder that we may or may not need to create. So if we're trying to create a new target folder, let's say the target tree as the name within the data folder, we can go ahead and first just check to see what current files exist within our data folder, and we can see we have target2 and target. But there's no target tree folder currently created. So let's say you were trying to use one of the quality control packages for your FCS files. Let's say you were running PicoQC, and you wanted to store any of those outputs to a particular folder. Now, if you didn't have that folder already existing, that script that you're running might error out and say target folder not found. But if you have a conditional set up that checks to see is that folder present, it's going to be able to actually check and see is it working, is it there? If it's not there, go ahead and create one. So basically what we can do is if we go ahead and make that file path, so in this case data is equal to target tree, that file path, if we pass it to the directory exists function, we can kind of see it's currently non-existent, so it's going to return to us a value of false. Now, if we then took that line of code that we just saw that's going to return as false, but we add that little exclamation point in front, we're basically flipping that scenario. So basically, what we're telling our if statement is, in a condition where this folder comes back and is being evaluated as false, flip it, which basically means this conditional now becomes active, at which point the dir.create function we had made will go out and actually create that particular file, that folder that we're interested in creating. And sure enough, if we run that line of code as it's currently written, we'll get a target tree folder that now is created and available to our local environment. All right. So at this point, we have FCS files within Target and Target2, as well as within our data folder. We can also need to copy copy FCS files to the target tree folder, but we're going to try to do so programmatically via R. And the way we do this is with the file.copy function. So the way the file.copy function basically works is what we're going to do is we need to provide two particular arguments. We need to provide from, which is going to be the list of objects that the list of files we're trying to copy to a different location. And then to, which is basically going to be the file path to the location we want to copy them to. However, if we try to run it as it is, with the existing variables and objects we have within our system, we're going to get back false, 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 basically meaning the 4 objects within infants, none of them were able to copy over successfully. And we get back an error message that looks like this. There was a problem copying the particular file name to that target location and then appending on the actual file name. So the actual issue is not so much where we're trying to copy it to. There is a data and there is a target tree folder. But if you look at it, it's trying to find a file name of just cell counts, etc., etc. But it's not actually including that these are not present within your working directory. They're present within that data folder inside your working directory. So at this point, what you need to do is basically we need to be able to update the list of files that we have to include the directory information so that when we're trying to copy over these files, they're able to successfully locate the files in the first place so that they can then be copied over. So as we see right now, we're working within our Git working directory, but we basically need to tell it these files are actually present within the data folder. And this is where that full.names argument comes in. So basically, full.names will also include all the actual folders between your working directory and that particular file of interest, and we'll just go ahead and create the file names with those set. So if you remember, for example, what we had previously, when we set full names as the argument to true, it then goes and evaluates it accordingly. And you can now see it's showing the full file path to these particular files, showing us data and then the name of the particular file set. And then at that point, we just have to repeat the steps we have already done. We need to filter for those INF infant specimens, which we can do via the same kind of logic for structure detect, because it's still going to be able to see the INF pattern. And at that point, we now have the infants value re-updated to also include the data part of the file path directory. And then at that point, we can then go and try file copy those particular 4 files that are now correctly specified as to where they actually are in relation to our working directory to our new target folder that we want to copy them to. And sure enough, if we run this code, we'll see all those files get moved to our target tree directory. Now, the main thing, just like everything we can do in R, if there's one way of doing one thing, there's always the way of doing the reverse. So just like there's a way of copying files to a particular location, there's also the option of removing files programmatically. But I won't actually go into how to do that today. The main reason is, unlike if you delete something in Windows or Mac or Linux and it goes to your recycling bin, if you delete stuff in R, it's kind of like permanently deleted, and short of going to a data recovery service, you're not going to get those files back. So especially for people who are just getting started learning how to work with file paths, I would rather not have you guys accidentally delete a folder that's very important to you just because the file path wasn't exactly how we needed it to be. But the actual function we may be using later on in the course is basically the unlink function. This is how we go and access the actual arguments as far as how it actually works. Kind of similarly, what we have been working with before we set the full.names argument to true was just the file name itself. But you notice for list.files, when we set full.names is equal to true, it gave us also the directory present within those same location. So what happens if we had only wanted to retrieve the file name and we only had a list of files that had been generated using the full.names is equal to true argument? Well, there's a function in R that's known as basename, which will basically clip off all the rest of the file path and just keep the actual file names. So if you were just like interested in the file names themselves, you'd just basically put that particular object with the file names present within base name, run it, and then you'd be able to evaluate it at that point. And similar thing, we've been working with the list.files, and so far we've used path, we've used pattern, and we've used the full.name arguments. We still had one additional argument we've been using as recursive is set to false. What happens if we set this to true while being actively working within our working directory? What we can see happen is basically the recursive equals true will mean it will search each folder that's present and then return the files within every single one of those folders. So in this case, when we set the recursive equals true argument, when we're using that same line of code we started with at the beginning, it now returns all the FCS files that are present within the data folder. It returns all those FCS files that are within the target2, and the target tree data folders. And then it also returns a bunch of screenshots I took when I was actually writing out this particular session. So you can see recursive equals true can be useful if you're just trying to get a list of everything that's present within a particular folder. The one thing I should caution you is you should make sure it's working within a small working directory, because if you accidentally run this on one of the very large files of your system, if you run it on your home directory on Mac or Linux, or if you run it on your C drive if you're on Windows, it's going to take a very long time to index and list all the files that are present on your particular computer. So use it with a little bit of caution just so you don't have to abort R as far as it's not working, it's kind of stalled trying to do all this effort. And finally, we're now towards the end of what we did as far as our workflow. We have made a lot of changes to our week 2 project folder. So this is where I will normally, in my day-to-day practice, try to make sure I save all the changes I have made to my code all those changes to those files and then back them up on GitHub so I can have them for next week, like worst case scenario, something happens to my computer between now and then, or I switch between computers and I need access to the code that I wrote this morning. And the way you would do this within Positron is, once again, you go to the far left to the Actions bar, you would select the Source Control tab. You'd select the little plus sign next to Stage Changes, which would add all the particular files, all the particular folders that have been updated between when you did that initial commit at the beginning of the session, at which point you could write a commit message, hit commit, and then push those changes to your GitHub account so they're backed up on the cloud so you're able to use them later on. All right. In conclusion, we have started introducing the concepts as working directories, file paths, and how we are able to use them to locate particular files that we're interested in. We've also gone through some of the more basic R terminology as far as how are objects and variables created, the assignment arrow, how to check the particular type, whether it's character, numeric, logical, for these particular values that are present within an object. And then we've also been playing around with file paths. So this will become quite useful as we go into next week because we'll be working with more with FCS files. And then we want to make sure we're providing just the FCS files to a particular function of like the flow core or the flow workspace package, because they would not know what to do with a CSV or with a folder unless we know how to designate them. And with that, I would talking what's going to happen next week. I feel very guilty at this point because I've subjected you guys to 2 weeks of mostly just basic R course knowledge, and I've only barely touched on the flow cytometry. So we're changing this up next week. So many of us worked quite extensively with FCS files, but not so many of us actually know what's within an FCS file. So next week we'll be taking a look inside the FCS file, kind of see what the major structural elements of an FCS file are, like where all these keywords we set in going. Can we actually look up the gain or the voltage settings that that particular FCS file had for a particular day? How do we get back the actual floor MFI values? How do we see if our user was running something on low, medium, or high acquisition speed? We'll be taking a dive at that, but then we'll also be building on some of the concepts we've worked on today in terms of basic R knowledge and building them into more structured objects like matrices and data frames, just so we have a little bit of a building block of what's going on. And with that, we're at the top of the hour, so I'm conscientious of your time. So thank you for everyone who has to leave now. As far as additional resources for this particular week, Riffamona's project, which is by Pat Schloss, I'm pretty sure he's at the University of Michigan, is a really good YouTube channel for all the basic R knowledge. So kind of like I refer everybody to University of Chicago YouTube videos when it comes to flow cytometry. He's the equivalent of anything when it comes to R. I tend to refer them to his channel because the odds are pretty high. He has covered the topic at least in some capacity, and he does very good explanations, and his zoom on his actual codes is very zoomed in, so it's quite useful. All the things that we've been working with today are in the context of what's known as a project-oriented workflow. Basically, we create projects and then keep all the code associated for that particular project within it. And then that's kind of what the article I list is kind of more of a description as far as like why we do that and not some of the alternative approaches you may encounter in some of the more other R videos. And then also, if my explanations were not quite great for some of what we covered today, I provided 2 additional resources as far as explaining file paths that are YouTube videos. Dr. Padillas is particularly good when trying to explain the nested folders concept. And as always, there's a couple optional take-home problems. You don't have to do these. You'll still be kept up. You'll still keep along with the course even if you don't have time to do them on a particular week. These are more aimed at trying to get you to explore outside of a particular curated experience, working more with your own data and kind of figuring out how to troubleshoot things as you go. So a lot of us as flow cytometrists are working with like a lot of external hard drives, a lot of USB sticks that we're plugging in to copy and transfer over files. And if you're like working on a Windows computer, unlike everything on your computer, which is under the C drive, these are going to be under like an F or a G drive, depending on how many USBs you have plugged in. So I kind of want you to be able to, like on this first problem, figure out how to get a file path to identify things that are on your external hard drive, and then attempt to copy over some of those FCS files from today to a target folder on your external hard drive. Just remember, you're just copying. There's no deletion. You need definitely to walk before you're able to run, at least in the context of R. And it looks like we had a small code glitch here. So both of these problems are like right on the same view. So the second problem is we kind of just used list files with the full names argument set to true. But I also just showed you base name, which is basically just going to retrieve the file names. In R, there's a couple functions that would just return the folders for those files that are present in the full file path name. I want you to kind of search the internet and see if you can figure out what the equivalent of base name is when it comes to those individual directory folders. And then finally, a lot of our packages will have internal FCS files that are used for the documentation. Oftentimes, these can be reached by the system file function. So basically, I know that FlowSum and Flow Workspace had at one point FCS files within them that were used for the documentation. So I'm kind of sending you on a goose chase to kind of see how do you navigate these file paths within the system.file to locate where these FCS files are within these particular packages. So these are the 3 optional take-home goals for this particular week. And with that, I thank you for your attention. And if anyone has any questions, I'll be in the chat for a little bit. And if not, the discussion forum will be open and we can touch back on any questions that you may have or do additional explanations throughout the week. And I thank you for your attention.